Hello, welcome to GCB 531. My name's Harold Reithman. I'm an associate professor of genetics, and I work at the Wistar Institute, where I've been involved in characterizing the subtelomere and telomere regions of the human genome. Today I'm going to talk about the history and the background of the Human Genome Project. The Genome Project is really a seminal human endeavor that that sets the stage, I think, for almost all of human biology from here forward. And what I hope I can convey in the talk today is a sense of how the Human Genome Project came to be, what are some of the key components of the Genome Project, and just to give you kind of an overview of how it developed over time. So in addition to providing the overview of the Genome Project, I'm also going to provide a brief overview of chromosome structure to get everybody kind of on the same page. I know many of you have different backgrounds, and I wanted to at least lay the groundwork for some of the very basics that are going to be necessary for understanding how chromosome structure and chromatin organization affects how the human genome was assembled. So let's start at the beginning. The key molecule, of course, for the human genome is the DNA molecule, and what you're looking at here is is a diagram of the human DNA molecule as deciphered by Watson and Crick in 1953. The key concept, several key concepts arose from this structure of this molecule. Among them is that it really defines the physical basis for all of inheritance. So this is the material stuff that gets transmitted from one generation to the next. The second key concept is that the structure of the DNA molecule itself provides an important clue for how it replicates. There are two backbone strands in the DNA molecule that are interconnected by bases, and the bases pair in a particular way. So if you have a single strand with bases, it automatically determines the opposite strand. And so by denaturing the DNA into two strands and then resynthesizing the complementary strands, you have a mechanism for DNA replication. The third important concept here is that the DNA in a cell is finite. And this is particularly important for the Human Genome Project because I think it was one of the key motivating features behind the project, in that since all of the information content required to build a cell or an organism is encoded in the DNA, and the amount of DNA in any cell is finite, if one could decipher the deoxynucleotide the DNA sequence of all of the DNA in a given cell or cell type or organism, you would have basically a database of information that encodes all of the processes that are important, that go on inside of the cell. So the idea that in one really big push you could get all of the information about all of the DNA in a cell could potentially be extremely important, because then you could just refer back to that information over and over again and in trying to understand what that information does. So in the human genome, the DNA is organized into chromosomes, 23 pair, 46 chromosomes, and that's shown in this slide. Once again, this emphasizes the finite concept. This is all there is in terms of the DNA that's encoding all of the functions that are required to go from a single cell to a complete human being. All of the proteins are encoded. All of the other messenger molecules are encoded by this finite set of DNA molecules. Once again, if you can understand exactly what the chemical basis of that DNA molecule is and get that information into a database, then one can go back and try to figure out what each stretch of DNA does and what its function in fact is in the cell or in the organism. The concept of genomics, although it's fairly new in terms of looking at the entire genome, it developed over a long period of time. And in just a real brief overview of some of the key highlights of genomics pre-1980s is listed here. Of course, you have the early elucidation of the laws of inheritance by Gregor Mendel, independent assortment, and so forth, and the development of the first genetic linkage maps by Drosophila geneticists in the early 1900s, followed by the physical maps, which were essentially cytogenetic maps of polytene chromosomes, where you could identify physically positions along Drosophila chromosomes through light microscopy. As I mentioned, the structure of DNA was a key breakthrough moment in the history of genomics by identifying the physical basis of inheritance in terms of the DNA, the structure of the DNA molecule itself. Molecular cloning was an important breakthrough that led to a lot of the technology that was used during the mapping and sequencing of the human genome. DNA sequencing, of course, is a critical technology that was developed by two different groups in the early to mid 70s. The method that eventually became the primary method for deciphering the human genome was developed by Sanger and co-workers. Human RFLP mapping. RFLP is restriction fragment length polymorphism mapping. This technique allowed individuals to mark segments of genomes with particular what were called RFLP markers, and by being able to identify variations in the DNA sequence, one could trace the inheritance of those pieces of chromosomes from one generation to the next. And by combining a large number of these markers with a large number of extended families, Botstein et al. Was able to construct a fully connected genetic linkage map of the human genome, which was an important advance, because once you had those marker maps, one could envision connecting the markers and isolating DNA that corresponded to individual traits that one knew was associated with particular markers. From 82 to 88, a lot of really important technical breakthroughs happened that led directly to the ability to embark on the Human Genome Project. One of the most important was pulse field gel electrophoresis by Schwartz and Cantor in 1983, and this permitted, for the first time, the ability to separate out very large segments of genomic DNA. And I'll get to that again in a moment. The polymerase chain reaction was another key technical breakthrough that enabled the chemical synthesis of DNA that was specific for any particular segment of DNA that one wished to isolate. Multipoint linkage mapping was developed by Lathrop et al. in 1984, and this is an extension of the techniques used in the RFLP mapping projects that allowed a more sophisticated generation of genetic linkage maps. Global physical clone maps were developed by several groups in the mid-'80s initially in model organisms, and these were really the first cases where entire genomes, in this case either of the yeast Saccharomyces cerevisiae or the roundworm C. elegans, the genomes were completely reconstructed from individual molecular clones. And obviously this is a key aspect in generating entire genome sequences. One first needed to map the genomes in segments of DNA that one could fully sequence and then reassemble. The first proposals to sequence the human genome were proposed in the roughly the mid '80s, initially by DeLisi, who was with the Department of Energy. The concepts that were driving the Department of Energy at that time were looking at the effects of radiation on human biology, and of course it was known that radiation affected DNA, caused mutations, and so forth. And this was viewed as a potential project that could be carried out most effectively in a large biology kind of environment, which was quite controversial at the time. Yeast artificial chromosomes were developed in the late 80s by Maynard Olson's group, led by David Burke et al. And these were really the first molecular clones that were capable of carrying very large segments of DNA molecules. And this was particularly important because all of the genome work so far had really been done in model organisms, where the total amount of DNA was quite limited. The human genome, on the other hand, with 3 billion bases, was 10-fold to 50-fold larger than these model organisms. And in order to really conceptualize how you're going to biochemically characterize all of the DNA and delineate all the sequences, it was important to think about it in terms of being able to isolate clones and characterize the clones one at a time using DNA sequencing. The automated DNA sequencers came online in the mid to late 80s. The first ones were developed by Lee Hood and co-workers. The culmination of all these different technologies led to some proposals to map and sequence the human genome, and the National Research Council in 1988 met and thought these concepts through and came up with a concrete proposal. So I mentioned pulse-field gel electrophoresis. I just wanted to show you a quick example of that. This was a huge advance over conventional gel electrophoresis in analyzing DNA molecules. In conventional electrophoresis, you were limited in the maximum size of the DNA molecule that could be separated conveniently on an agarose gel. And by altering the orientation and directions of electric fields, it was shown that you could actually separate much larger molecules than had been separated previously, all the way up to megabase-sized pieces of DNA. And this enabled the mapping of very large chromosome regions, and also enabled the development of large insert cloning technologies, in particular yeast artificial chromosome cloning and bacterial artificial chromosome cloning. The polymerase chain reaction was a method that enabled the in vitro synthesis of double-stranded DNA from any target DNA that you had the sequence for. And this worked by starting out with 2 short pieces of DNA called oligonucleotides that were oriented in opposite directions, and annealing those to your target sequence and adding the appropriate polymerases and chemicals under the appropriate conditions. Both oligonucleotides would extend and complete the first strand. Then you would denature, re-anneal the oligonucleotides, do it again, and so you'd get an exponential synthesis in the target DNA molecule that was in between the oligonucleotides. And this provided an important method for amplifying pieces of DNA of particular interest. It also enabled the conversion of DNA markers into a very convenient format for developing physical maps and genetic linkage maps. So by 1988, there had been a fair amount of consensus reached on what the Human Genome Project might look like and whether it should be initiated. And of course, the conclusion was that it should be initiated, that it was an extremely important piece of information to have a complete data set of all of the human genes and a complete sequence of the human genome. In order to get there, however, it was recognized that there are a lot of steps that had to happen. Technology development had to be improved in almost every phase of the project. The the early goals would be focused mainly on making maps and clone collections and developing the technologies that would be required for sequencing. A second really important aspect of this report was that it emphasized the need to sequence the genomes of model organisms in parallel with the human genome, because once you have the human genome, the model organisms were really going to be critical for trying to understand the function of the human genome because of the evolutionary relatedness of the model organisms to the human genome, in combination with the experimental facility of model organisms. One could take a particular gene or segment of the human genome, look at its evolutionarily conserved homologue in one of the model organisms, and systematically mutate the gene in the model organism in order to figure out what that particular gene actually does functionally. So a really important concept in the Human Genome Project is the concept of maps. And maps are composed of two components, landmarks and fragment ends. The landmarks correspond to short segments of DNA that can be recognized either by hybridization technology, using DNA probes, or by using the PCR technology that I just described, using what were called sequence tag sites, which are essentially PCR assays that are unique for a specific short segment of DNA. The generation of fragment ends can be done in a number of different ways depending on the kind of map that one is constructing. They can be generated by cutting the DNA with a particular restriction enzyme, which is a DNA endonuclease that recognizes specific stretches of bases along the DNA molecule. They can be generated in vivo, meiotically, by using the meiotic recombination system of the particular organism that you're studying. And then you map the DNA landmarks relative to where those meiotic breaks occur. Radiation could also be used to generate breaks, usually in the context of a genome within a cell line. And by cloning the DNA, you automatically create two fragment ends, one at either end of the clone. And those can also be used to fragment the genome and then use the landmarks to piece together those fragment ends. So if this is a DNA fragment, for example, and you have these series of landmarks, if all of these landmarks can be recognized by this single DNA molecule, you know that they're all present within whatever this distance is, which corresponds to the size of the DNA molecule. If you have a series of DNA molecules and some, but not all, of these very same markers recognize this second DNA molecule, and you have additional markers that recognize the molecule that do not recognize the first molecule, then you can place these markers roughly relative to these DNA fragments and identify this segment as an overlapping segment of DNA between the two fragments, and so on and so forth. You have additional DNA fragments that recognize some but not all of these markers, and have additional markers that it recognizes. And by continuing to grow these different what are called contigs, you can eventually cover the entire genome with these different kinds of maps. So I mentioned briefly sequence tag sites, and this was actually a pretty important advance for logistical reasons as well as for conceptual reasons. Much of the mapping had been done using cloned DNA fragments called probes, and these probes were distributed from laboratory to laboratory, and in fact large repositories of probes were created explicitly to be able to distribute these pieces of cloned DNA fragments, which had to be grown in live bacteria and repeatedly purified by different laboratories. Sequence tag sites made use of PCR technology to simplify these DNA landmarks or these DNA probes. And essentially what one could do is make a pair of oligonucleotides that corresponded to any given DNA probe and put that in a computer database. And any lab that wanted to create the landmark would simply need to download those sequences and the spacing of those oligonucleotide sequences in the appropriate PCR conditions, and you could regenerate the PCR assay in your own laboratory without having to go through all of the hassle of distributing clones and making sure it's the right clone and so on and so forth. And so this really enabled the mapping technologies and propelled the projects forward. In addition, because you have the same specific markers, you can use these PCR-based markers for lots of different kinds of maps, for genetic linkage maps, for radiation hybrid maps, for clone-based physical maps. And so the different map types could be connected by virtue of these identical sequence tag site markers or STS markers. And so the concept of STS maps became an important concept for genome mapping and sequencing. An important kind of map that was developed, I've already alluded to, the genetic linkage maps. And these are created on the basis of meiotic breaks. And during meiosis, homologous chromosomes exchange information along the chromosome, and the sites of the DNA breaks that correspond to these exchanges are called meiotic breaks. And one can take STS markers or other kinds of markers and determine which pieces of the chromosomes that are exchanged from going from one generation to the next. And if you have, for example, a pair of markers that are on the same piece of exchanged chromosomal DNA, you know that those markers have to be spaced within that meiotic interval. And just as I mentioned earlier with other kinds of maps, you could then increase the density of markers along the chromosome and increase the number of meiotic breaks by looking at additional families and additional pedigrees. And you could in fact build and construct entire genetic linkage maps where the markers that are closest together are almost always mapping to the same pieces of DNA that get transmitted in these meiotic chromosome fragments. And you could, as before, you could identify intervals of chromosomes where some markers are on one side of the breakpoint, other markers are on the other side of the breakpoint, and by doing that, you can actually order the positions of these linkage markers along the chromosome to create genetic linkage maps. An additional important aspect of genetic linkage maps is that because they're created in vivo, you can also map traits on genetic linkage maps. So if you have a mutation that's known to cause a disease, and you know it's transmitted from generation to generation, you can then look for markers that are tightly associated with the transmission of that trait from one generation to the next. And because those markers now have a concrete position along the chromosome, you can take a marker that lies on one side of the mapped trait and a marker that lies on the other side of the position of the mapped trait. And by cloning all of the DNA in between, you know that you must now have cloned the DNA that actually caused the trait that you're interested in. Radiation hybrid maps are similar in principle to genetic linkage maps, except that instead of meiotic breaks, one generates the breaks using X-rays, and you can generate the frequency of the breaks by adjusting the energy of the X-rays. Once the chromosomes are fragmented, they're recovered in what are called somatic cell lines. And once again, if two markers are very close to each other on the chromosome, they'll almost always be recovered in the same radiation hybrid-induced DNA fragment. If they're farther apart, they'll be randomly distributed amongst the different recovered fragments. And using the same principles, the genetic linkage map, you can then deduce which pairs of markers are very close together, and looking at overlapping sets of RH fragments and RH markers, you can then construct an entire map of RH markers along the chromosome. Clone-based physical maps are really the feature of mapping that led most directly to the generation of DNA sequence. And this is because they're derived from pieces of the chromosome itself, and you actually recover clone fragments, which can then be characterized further all the way down to the DNA sequence level. And the principles is what I mentioned earlier, except that instead of breaking the chromosome into fragments using one of the other methods, in this case you're creating fragment ends by virtue of the cloning process itself. So with each insert that you clone, you have a limited size fragment. Typically, that fragment is defined in length so that you can map your landmarks to those different clones, and by identifying pairs of markers that lie within individual fragments and entire sets of markers that identify fragments that are covered by some markers on one fragment, other markers on another fragment, and the shared region by a third set of markers that cover both fragments. You can build up overlapping sets of clones and ultimately come up with a what's called a minimal tiling path of clone fragments that reconstruct the original chromosome region that was cloned. So based on these early mapping technologies and the development of the bioinformatics and the marker systems by about 1990 or 1991, the initial 5-year plan was set forth for the Human Genome Project, and it focused, as I mentioned before, on maps. The idea was to get fully connected linkage maps of the human genome, fully connected physical maps in the sense that you would have a clone-based physical map with markers at least 100 kilobases apart or closer, and this was a size that would permit overlapping contigs of yeast artificial chromosome clones all the way along a chromosome. Sequencing technology, it was recognized, had to be improved, so the goal was to drive down the cost, increase the efficiency of DNA sequencing, and to sequence model stretches of human chromosomes to get a sense of what the DNA sequence organization actually looked like to help guide future sequence development efforts, sequencing efforts. Model organisms, the goals were to generate genetic maps of the mouse and to generate 20 megabase stretches of sequence from a number of model organisms in the context of technology development. Informatics, it was important, and it was clearly recognized early on that databases, software, algorithms needed to be developed in order to analyze the sequence and enable the assembly processes to go forward. An important aspect of the Human Genome Project, which was actually fairly unique, is that it was recognized early on that this kind of information was going to be really important and different in key aspects. In particular, if people had access to their genetic information, there were certainly privacy issues involved because there would be a possibility that you might be able to use it to predict disease, predict health, predict different aspects of susceptibility and risk to lots of different kinds of situations. So, for example, if a health insurer found out that you carried the gene that predisposed you to Huntington's disease, they might, in fact, reject your application for insurance. And it was recognized that for the Human Genome Project to reach its fullest potentiality, it was really important from a policy perspective to come up with ideas and strategies to make sure that it didn't in fact hurt individuals to know their genetic information. And so about 3 to 5% of the Human Genome Project's budget was dedicated to the ethical, legal, and social implications of actually acquiring the human genome sequence. And that turned out to extend throughout the entire Human Genome Project and beyond. And it's played a very important role in healthcare legislation, in requiring insurance companies to not reject applicants on the basis of their genetic background. So just 2 years into the first 5-year plan, a second 5-year plan was generated primarily because of the extremely rapid progress in physical mapping, genetic linkage mapping, and also DNA sequencing technology. And that second-year plan is illustrated here, where many of the goals that were outlined in the first-year plan were already achieved by 1993. And so a new set of plans were set forth that included, in addition to the STS maps and the linkage maps, the identification of all of the genes in the human genome, and also very significant, substantial improvements in DNA sequencing technologies. Automated sequencing on slab gels had now become commonplace, and it was recognized that there was a huge window for additional improvement in throughput and efficiency in genome sequencing. That DNA sequencing improvement curve is shown here, where you see just a trickle of sequences coming in the mid-90s, and then a sudden ramp up in the late 90s. Much of this ramp up had to do with the transition from slab gel electrophoresis to capillary gel electrophoresis, and this enabled enhanced throughput as well as an increased accuracy in genome sequencing, to the extent that you could now have highly automated production-style DNA sequencing that was carried out in these large DNA sequencing centers, in this case at the Broad Institute, where you had your chemical reactions carried out in 96 well or 384 well plates, and robots automatically loading the capillary electrophoresis instruments, and computers collecting the data right off of the sequencing instruments. And this automation really propelled the sequencing projects forward, and made the key transition from mapping to large-scale sequencing possible in the late 1990s. An additional important factor was the competition that was now emerging from the private sector, initially in the form of Celera Genomics. And here the Celera group suggested that one did not necessarily need to have a completely mapped genome in order to completely sequence it. They suggested that with the improved sequencing technologies and improved computational methods, you could simply fragment the entire genome, sequence all of the pieces, and put all of the pieces together into a complete genome. The competing view and this was championed by the public consortium of genome sequencers, was that we really didn't know enough about the structure of the human genome to make that leap of faith. And in order to really make sure that we achieve the goal of having high-quality sequence all the way across the human genome, we needed to take it step by step and continue with the clone-based project and sequence basically one clone or small groups of clones at a time in order to piece together the human genome. And these two concepts led to two competing papers that came out with initial draft sequences of the human genome in 2001. The first from the International Human Genome Sequencing Consortium, the public sequencing group, and the second from Celera Genomics. Both draft human genome sequences, which I'll discuss in detail in an upcoming lecture, turned out to be quite similar in content, but the strategies used to generate them were significantly different, although there were, in fact, more similarities than differences, and some of the differences were primarily semantic and were not, in fact, huge differences at all. So from the initial draft sequence to the finished sequence of the human genome involved a lot of hard work by a lot of sequencing centers, and this was primarily because of the complexity that repeat sequences conferred and was needed to be overcome in going from what's called a draft sequence to a complete finished sequence. Once the finished sequence was complete, the Human Genome Project per se officially ended, but the work on genomics and the sequencing technology, as well as sequencing applications, has continued to the present day, and the basis for this was really laid out in 2003 in a paper that came out in Nature, describing the role of the human genome reference sequence in all of these future kinds of applications that would base their efforts on attaching function to human sequence, connecting health-related diseases to the genome sequence, and developing technologies that would interface between all these different aspects of the human genome sequence. And some of the ideas at the end of the Human Genome Project included an effort to identify all of the functional components of the human genome, and this was known as the ENCODE project, and it's still ongoing, in fact. And I'll also discuss some of these large-scale genomics projects in an upcoming lecture. So some of the other aspects of how the reference sequence would lead to improvements in understanding biology were the HapMap project, which was an effort to understand the variation in the human genome, an effort to understand variation across species and the mechanisms underlying variation, and of course the continuation of the ELSI project. Technology development was still ongoing and is still ongoing at a very rapid pace. Sequencing technology has continued to evolve, as you'll see. Methods to detect genetic variation proceeded from the detection of known variants to the generation of a large-scale database of known variants and technologies to type those variants in lots of genomes, to the current state, which is basically determining variation by resequencing entire genomes. Genomics parts lists needed to be developed. What are all of the genes in the genome? What are all the functional elements in the genome? Proteomics, pathways and networks, all of these aspects of genomics have continued their development since the Human Genome Project was completed in 2003. In 2003, we were also left with some sort of overarching goals that we thought could lead to quantum leaps in our ability to understand human genetics and biology. And one is to, of course, lower the cost so that we could get much larger amounts of variation information, and we could get complete information on the mutational status of individual humans. Part of this was the ability to sequence DNA at costs that would reduce the cost of genome sequencing to basically a kind of a medical test. So one could envision having as part of your medical record your particular genome sequence. And in the case of disease, perhaps cancer, for example, you could analyze multiple genomes, multiple cancer genomes as the disease progresses and get information on how best to treat that disease. Other important goals were to attempt to look at things at the single cell level and try to simultaneously monitor the cell state in terms of looking at all of the proteins in a single cell in a single experiment. Many of these have not been achieved yet, but some of the sequence-related ones, in fact, many of those goals have been achieved or are getting close to being achieved. So chromosome structure, I wanted to give you a quick overview of chromosome structure, and in particular how it relates to the genome sequencing and assembly aspects of the Human Genome Project. So I showed you before metaphase chromosomes. This is the sort of the unit level organization of the human genome. Already you have it divided into 23 different segments, each segment being, of course, a complete chromosome from one end to the other. And in fact, the just having the ability to map sequences to these karyotypes using fluorescence in situ hybridization methods turned out to be a very important way to connect small, relatively small scaffolds of sequence to the actual genomic locations along the chromosomes. The concept of a gene we haven't really talked about specifically. I thought I'd just mention that here. It has several different kinds of definitions depending on at what point in time it was being discussed and what else was known about the chemical basis of the gene at that time. Initially, it was purely a genetic concept. It was thought of as a piece of a chromosome upon which mutation might occur. It would have some sort of functional effect on the organism. The physical concept that evolved after DNA was worked with was that it corresponded to a string of nucleotides on a chromosome that encoded a discrete function, typically through the action of a protein that it encoded. Later non-coding RNAs were discovered. Actually, the first non-coding RNAs were discovered long ago. They were the ribosomal RNAs and the tRNAs. But additional non-coding RNAs have been discovered that also, upon mutation, confer phenotypes, and so these are also thought of as genes. Heterochromatin is a class of DNA that has particular characteristics. The heterochromatic DNA typically occurs in condensed chromosome regions. They typically have a particular structure that's composed of tandemly repeated DNA elements. Sometimes these elements are repeated for very long stretches, millions and millions of base pairs. Because they're large and they're tandemly repeated, they're very difficult to clone and assemble. Bacteria as well as yeast don't necessarily handle these large tandem repeats particularly well. They tend to delete out upon DNA replication in these clones, in these organisms. And even if you can clone and sequence them, they're very difficult to assemble uniquely because they all have almost exactly the same sequence. In addition, heterochromatic sequences are typically gene poor with low transcription levels, and in addition, they're enriched near the centromeres and at the telomeres of many organisms, including humans. Here's an example of the sequence structure of a heterochromatic region, in this case a centromeric satellite, where you have, with the small red arrows, you have short stretches of tandemly repeated DNA segments, and these can be grouped into larger sub-repeats that encompass larger and larger regions. And as I mentioned, these can extend over very large distances, millions of bases and more. Euchromatin is the rest of the genome, the part of the genome that's not heterochromatin. It's highly variable from region to region. It covers a wide array of DNA sequence types and sequence organizations with lots of different percentages of interspersed repeats and different kinds of duplicated genes and so forth, but conspicuously lacks these large segments of tandemly repeated DNA. Most of the genes are located in euchromatin, very unevenly distributed throughout the euchromatin. Some regions of euchromatin have a very high density of genes. Some regions have a very low density of genes. Most of the euchromatin is accessible to cloning and assembly. So when we talk about the human genome and the human reference genome, we're really talking about the sequence of the euchromatic region of the human genome. This is a segment, an annotated segment of a euchromatic region. The blue gene structures are shown at the top, and below that is the same region showing all of the interspersed repeats, and this is to illustrate that about half of the euchromatic region is actually comprised of these interspersed repeats, and they're an important component of the euchromatin involved in almost every aspect of euchromatic biology. Segmental duplications are present throughout the euchromatic regions as well, and these are defined as large segments of recently duplicated DNA greater than 1,000 bases in length and greater than 90% nucleotide sequence identity with at least one other duplicated segment in the genome. These regions are enriched in the pericentromeres, which are the euchromatic regions right next to the centromeres, and the subtelomeres, which are the euchromatic regions right next to the heterochromatic telomeres. They tend to be associated with large polymorphisms, so these duplicated segments differ in copy number and size from individual to individual, and they tend to also be associated with deletions that often take out adjacent genes, delete out adjacent genes, and cause disease. This is an example of the segmental duplication organization of human chromosome 7, which is shown in the central part of panel A. The segmental duplications that are duplicated within chromosome 7 are shown in blue. Those duplications that are present on non-chromosome 7 places in the genome are shown in red. And this illustrates that these are fairly abundant elements in the genome. They comprise about 5% of the euchromatic region of the human genome. And it turns out, as you'll see later, that they pose some problems, particularly in the assembly of the finished human sequence, and often they are vastly underestimated in draft genome sequences. So very briefly, onto chromosome function. Chromosomes are the units of heredity, so that one piece of DNA goes from one end of the chromosome to the other. So how they're replicated and divided amongst the daughter cells is, of course, critically important. And I wanted to touch on some of the key functional elements of chromosomes quickly. An important functional element, of course, is the replication origin. And in large genomes, such as the human genome, the chromosome, the initiation of replication starts at multiple sites along the chromosome called replication origins. And as the DNA is replicated, these bubbles form along the chromosome and eventually merge until you get two completely new double-stranded DNA molecules from your original DNA molecule. The DNA itself is replicated through a leading strand where the polymerase uses one of the denatured strands as template to make a new strand. And in the lagging strand, the synthesis is in the opposite direction, and it's synthesized in a series of short DNA fragments called Asaki fragments, and these are primed by short pieces of RNA. But what you get at each replication fork, which is what this is representing here, is the DNA as it unwinds, it's being replicated as it's being unwound, and so you get the replication fork moving through the double-stranded DNA molecule and leaving behind two daughter molecules. Centromeres and telomeres are the other key functional components of chromosomes. The telomeres are the tips of chromosomes, and the centromere is the constriction along the chromosome that is critically important for the proper segregation of the chromosome to the daughter cells upon replication. So centromeres can form anywhere along the chromosome. If the centromeres are at the very end of the chromosome, they're called telocentric chromosomes. More typically, they're not at the very end; they're somewhere along the body of the chromosome. They form the attachment sites for microtubules, and during the replication process, the chromatids are pulled to one daughter cell or the other through the attachment of the microtubules to the centromere, ensuring the proper segregation of the genomic material. Now, the sequence organization near the centromere is particularly complex, and this is an important feature that will impact the human genome assembly. As I mentioned before, there's heterochromatic centromeric DNA, and this forms the core of the centromere, and this region is typically absent in genome assemblies. Then you have the typical euchromatic sequences that have the usual distributions of all of the other gene encoding regions as well as the interspersed DNA. In between, you have this boundary region where you have a mix of heterochromatin-like material along with euchromatic-like material, and very often segmental duplications are also particularly abundant in these boundary regions. So many of the gaps in the human genome reference sequence actually occur in these boundary regions that are in the pericentromere regions. Telomeres form the tips of human chromosomes. They're absolutely required for chromosome replication and the stability of chromosomes. If the telomeres are lost, then the DNA looks to the cell like a piece of broken linear DNA, and the cell treats it as such, and it tries to recombine with other pieces of linear DNA and causes genome instability. So you have to have intact telomeres for appropriate replication and stability. The telomere itself is made up of a simple repeat sequence that's maintained by an enzyme called telomerase. And the telomerase enzyme recognizes this particular motif, and through the action of a ribonucleotide, RNA component of the telomerase enzyme, lays down additional copies of this and synthesizes new telomeres. Subtelomere regions are the regions adjacent to the telomere, and similar to the pericentric boundary regions, subtelomere regions have interesting features in lots of different organisms. They tend to be rapidly evolving, meaning that they duplicate. They tend to duplicate a lot and delete a lot and be polymorphic. And so gene families that are embedded in these duplicated sequences have a chance to very rapidly shuffle exons and make new combinations of potential genes. While the precise sequence of the subtelomere is completely different from organism to organism, the organization of the duplications shows some similarities, and that's illustrated here. And the importance of this for genome assembly is that, as with the pericentromere regions, the subtelomere regions can be especially difficult to put together into a finished form. And in the case of draft genome sequences, they're particularly susceptible to gaps and overcollapsing and difficulties and errors in the genome assembly. So the replication of chromosomes occurs in an ordered fashion in the cell cycle, and the division of the cells also occurs in a very orderly fashion through mitosis. Meiosis is the specialized division of germ cells that creates meiotic recombination and ultimately leads to haploid germ cells. So the cell cycle is illustrated here. It goes through several distinct phases, G1, S, and G2, and M. G1 is a resting pre-DNA replication phase. S phase is the DNA synthesis phase. G2 is a post-synthesis phase where the DNA is now has a copy. Each DNA strand has a copy of itself, so you have a 4N DNA content. And mitosis is when the cell actually divides and produces two daughter cells. The entire G1, S, and G2 phases are also known as interphase, which is that part of the cell cycle where mitosis is not occurring, and of course M is when mitosis occurs. Mitosis and meiosis share a lot of similarities, but they have key differences that it's important to understand. In mitosis, you start out with one set of homologous chromosomes. You go through S phase to produce two sets, and each set, I'm sorry, each set of DNA of duplicated DNA is present in what's called a chromatid. So you start out with a single chromatid. The chromosome is replicated, so you have two chromatids joined at the centromere. At mitosis, those chromatids separate into daughter cells, and so you end up with exactly what you started out with, a diploid daughter cell with a genome that's basically identical to what you started with. In meiosis, you also undergo a round of replication, end up with sister chromatids on each chromosome, but then you go through a meiotic prophase. And during meiotic prophase, which is illustrated here, where you see the chiasma, the chromosomes cross over and the chromatids exchange DNA information with each other. At the first meiotic division, the homologous chromosomes actually separate, and so you get a decrease in chromosome number from 46 to 23. And in meiosis 2, each of the chromatids separate and leave you with a haploid cell, and these haploid cells are the gametes. So even though you undergo one round of DNA replication, you actually undergo 2 rounds of cell division to leave you with the haploid gametes. It's important here as well that in terms of genetic linkage maps, that the chromosome exchanges that occur during meiotic prophase are the basis for genetic linkage maps.